years. So I will switch gears from physiology to developmental biology, as I am a developmental biologist at heart, and we are very much interested in how lymphatic vessels form. And we use zebrafish as a model system to study this question. And what we have been doing is utilizing a lot of live imaging to try to understand the dynamics of the developing lymphatic vessels. And of the purpose of this talk, I will give you a little bit of introduction into why we use zebrafish and a little bit more introduction to lymphatic vessel development and their function, since I'm aware this audience is quite broad, and I hope I'm not gonna too generalize my talk for you guys, so please bear with me. So why do we use zebrafish? This is a very useful model in developmental biology, mostly because the embryos are developing outside of the mother, which means that we have access to the developmental processes as they happen from sort of set, go, start. And we can really follow them throughout the formation of whole tissues. In addition, these embryos are transparent, so with you can utilize transgenic lines to visualize tissues of interest. And as you can see here at the bottom of the slide, at 5 days post-fertilization, most of the organs are being formed. So the digestive system, brain tissue, eyes, and so on are already established. So you can imagine this is quite a rapid development. An additional great advantage of zebrafish is also that they produce quite a large number of embryos, so they're quite useful for drug screening, and these type of screens are becoming much more cost effective. So here is an example of 2 transgenic lines. The middle panel shows transgenic line that highlights a heart by 2 different fluorescent proteins, green and red, which are being driven in the myocardium and endocardium. And at the bottom is a you can see the vasculature of 5 days post-fertilization embryo. And you can, using this combination of transgenic line, one can distinguish between the arterial, venous, and lymphatic vasculature. So you can imagine this is quite powerful to really see how this, how the development happens as a whole across the whole developing embryo. And of course, we have a number of transgenic lines that we can use in the lab to visualize lymphatic vessels. And this is an example of one of these transgenic lines that drives PROX1 expression, GFP expression by PROX1 regulatory elements. And here you can see the main lymphatic vessel, which is thoracic duct, associated vessels. And PROX1 is a transcription factor that is also present in other tissues, such as neural tube. You can see it here, as well as in the gut. And this is an example of a different transgenic line, again, highlighting the lymphatic vessels here in the face and also across of the body. So you can see the whole network of the lymphatic vessels at 5 days post-fertilization. And this transgenic line is also expressed in the venous endothelium here, this very thick vessel at the bottom. So these are sort of the tools that we use in the lab. And so why do we study lymphatic vessels? As we all know, there are 2 vascular systems that span of our body. One is cardiovascular network, which is a closed circular system. And as we all well know, it's essential for distributing the nutrients as well as oxygen throughout our body. And although it's vital for our survival, actually it's not such a perfect system, as it is quite leaky. And this is where lymphatic vessels come in play. And they actually collect this fluid that leaks out of the cardiovascular system and brings it back. So therefore, lymphatic vessels are built in a sort of unidirectional system of vessels. These are it's an open system and not a closed circular system. So there are clear differences between these 2 vascular networks. So what is the function of lymphatic vessels? I already mentioned it's really important for maintaining the tissue homeostasis, fluid balance, but it's also important for immune trafficking of the immune cells, for the immune tolerance, and absorption of the fat in the gut. And this is done by specialized lymphatic vessels called lacteals. And this network of vessels is organized in hierarchical manner, where the blind endel capillaries is the place where the fluid is being absorbed, and fluid then is being carried out through collecting vessels and also the thoracic duct. And as you can see in this image, lymph nodes are also part of this network, where of course a lot of immune responses are taking place. So, of course, there's a number of disorders which are associated with lymphatic vessels. And here it's one example of the most studied disorder, which is lymphedema. And this disorder arises due to the disrupted function of fluid absorption by lymphatic vessels. And as you can see here on these pictures, there appears accumulation of the fluid in the extremities. And these defects could be primary due to genetic mutations, and usually this arises during the development, still during the embryonic stages. But also lymphedema can be acquired due to trauma or injury. And here it's an example of a patient who underwent surgery for breast cancer, where lymph nodes were removed. And actually 1/3 of patients undergoing this type of surgeries end up with lymphedema in the arm of the affected side. So there are many reasons for lymphatic disorder to arise, and besides lymphedema, there are many other lymphatic related disorders due to other functions the lymphatic vessel has. And just to highlight why zebrafish is quite a powerful model to study lymphatic vessels formation and its relevance to human diseases, I think this is a very elegant example what really highlights the potential of zebrafish. So this study is relatively recent, and it has a group of patients was identified that had a mutation in the RF gene. This mutation was a gain of function mutation, which led to the sort of disruption of lymphatic vessels, where they became expanded and more leaky, and they weren't the fluid wasn't distributed as it should throughout the body. And these patients weren't responding to the current therapy that normally patients with such a lymphatic anomaly undergo. So what these researchers have done, they have generated a zebrafish line where they introduced this mutation the same mutation in the RF gene ectopically in the lymphatic vessels. And you can clearly see that this thoracic duct, the main vessels, is being really enlarged compared to the control overexpression of the wild type form of this gene. And then they conducted a small screen looking for MEK inhibitors in order to find one that would block this phenotype. And they successfully found a candidate inhibitor. And remarkably, when they have treated this patient with this drug, there was a really remodeling and recovery of the lymphatic vessels, both in the lungs, where the predominantly phenotype were, but as well as in the extremities. And I think this is a really powerful study that highlights all the advantages of using zebrafish model. So just to summarize this sort of introductory part of my talk, I hope I convinced you that zebrafish is an excellent model to study lymphatic vessels, but also gave you a bit of a feeling of different lymphatic pathologies and why this is relevant. So now I will dig in a little bit more to the research that we are doing, and I will share with you some of the preliminary data that we have in the lab, and this is also available on the sneak peek in case you want to read further after my talk. But just to tell you a little bit how lymphatic vessels develop in zebrafish. So we know that there are these progenitors that sit in the horizontal myoseptum, and they start migrating up and down in close proximity to the arteries and give this primary lymphatic vessel network. And they undergo, of course, specification, migration, and proliferation. And purpose of my talk, I will focus only on migration today. So we also know that these progenitors are derived from the posterior cardinal vein. They initially induce PROX1 expression, undergo a cell division event where only one daughter cells maintain high expression of PROX1, and in response to the VGFC signaling, migrates out and give rise to the further progenitors of lymphatic vessels. So in the lab, we are very much interested in how migration of these cells is regulated. So how these cells that sit in the horizontal myoseptum migrate up and down and give rise to this big network. And other projects running in the lab are concerning the proliferation. So what we know is that the migration of the lymphatic endothelial cells is guided by arteries, as well as by chemokines CXCL12 and CXCL4. And when I was still a postdoc in Ben Hogan lab in Australia, we actually found a mutant in a forward genetic screen that lacked lymphatic vasculature. And we were quite excited when we found that the mutation in this mutant underlined a gene called mafba. And this is a transcription factor. And thus far, we didn't know many transcription factors which are important for migration of lymphatic endothelial cells. This transcription factor previously has been shown to play a role in other developmental processes, such as hindbrain boundary establishment, also pancreatic and hematopoietic lineage differentiation. So the work during my postdoc has uncovered, using the time-lapse imaging, that this gene is important for cell migration. As you can compare the sibling situation with the mutant, you can see that these progenitors in the wild-type situation migrate rapidly up and down, whereas in the mutant situation, they're very much stuck. And this work, we found that this gene mafba is actually downstream of VGFC signaling pathway and SOX7 and SOX18. But one thing that was puzzling us quite a lot, we know that in zebrafish, very often there are paralogs of genes due to the genomic duplication. So there's not only mafba gene in zebrafish, but there's also mafbb. And when we look by in situ hybridization of the expression of mafbb in mafba mutants, this expression has been maintained, suggesting that actually there might be some compensation in the phenotypes, and that might be and that potentially the double mutants could have much more severe phenotypes. And we were quite surprised that when we generated the double mutants, we actually didn't observe larger severity of the phenotype. So when we quantified the lymphatic vasculature in the mafbb mutants alone, we didn't really observe any phenotypes. And as I said, when we looked at the mafba and mafbb mutants, the double mutants, the severity phenotype compared to mafba mutants has not been more dramatic. So we looked also at the onset of the phenotype, and we haven't seen any differences. So that puzzled us, but the story hasn't finished there, since I'm giving this talk. So there is more data, which actually came by a big surprise. And as I showed you at the beginning, the lymphatic network, it's not only along the trunk of the fish, but also the facial lymphatics, or the face is full of lymphatics as well. These lymphatics develop also through quite rapid migration and proliferation. And when we looked at the double mutants, mafba, we actually saw morphologically distinct lymphatic vessels compared to single mutants or the sibling controls. Further on, the number of cells that comprising this lymphatic vasculature was also slightly reduced compared to the single mutants alone, suggesting that these two paralogs actually play a topographically distinct roles, and the compensation only occurs in one region of the embryo, not the other. And that really surprised us, because such a phenomenon hasn't been described yet, at least not in the vascular field. So we wanted to know where these phenotypes arise and what's going on wrong. So first we look at the specification of this vessel in the face, and when we compared all the mutants with our siblings, we actually did not see any differences in specification, suggesting that similarly, like in the trunk, these genes play a role probably in later stages. We look more carefully at the morphology of these vessels, and clearly they were dramatically disrupted. where they seem to be sort of sprouting in many different directions instead of forming this very cohesive tissue comprising of just a handful of vessels. Recently, there has been described that there is a valve region in these facial lymphatic vessels that is important for functionality of the vessels, and this has been disrupted in these double mutants. So was the functionality of this vessel. So when we injected the dye into the interstitial space in the wild type or the double mutants, we observed that there was no dye accumulation in the mutant embryos after 3 hours post-injections, when there was dramatic increase of the dye accumulation in the wild type vessels, suggesting that indeed there is these vessels are being functional. So then we went on to do time-lapse imaging to see whether these phenotypes arise to problems in migration. And when we looked in the wild type, we could see how these lymphatic cells migrate quite nicely in very directional manner. However, as you can see here in the mutants, they are sort of stuck. They try to come out and try to migrate. They throw these phylopodias, however, they don't, however, they don't really progress much further. And when we quantified these phenotypes, we saw indeed that there is that these cells don't progress as far compared to the sibling cells, and the mutant. However, the mutant cells, the velocity of the movement of the cells has not been different. We further looked more carefully at the directionality of this migration, and what we observed is that the sort of collective migration of all the cells was disrupted, and the cells were migrating much more ventrally. When we divided the cells into the leading and the following cells, we could see that the leaders actually are trying to walk or migrate more posteriorly, and they sort of lose the sense of directionality, suggesting that these genes together are required for the directional migration. So with that, I would like to conclude, and I hope I have convinced you that these two genes are required for the formation of lymphatic vessels, but only in the facial lymphatics. What I haven't shown you, what we also further showed in the preprint, was that these two genes also respond to the VGFD signaling, and they're important for the fine-tuning of the migration. And as I said, the biggest surprise for us in this story was that there's this topographical difference in requirements of these genes. So with that, I would like to thank my lab members, and in particular Hannah Arnold, postdoc in the lab, who started this work, and then Virginia Panara has helped her to bring it to more of a complete state. I also would like to thank Ben Hogan and many members in Ben Hogan Lab, where the Mafb story started, and who contributed to initial discovery of the Mafb mutant. And of course, I would like to thank my funding and the fish facility for taking care of our fish. And with that, I'm happy to take questions. And if you would like to contact me afterwards, here are the details how you can reach out to me. Thank you very much for your attention.
Thank you very much, Kaśka, for the great talk. We have one question from Jonas. How are the migrating lymphatic cells able to migrate only on the vascular scaffolding and not elsewhere? Do they sense the network?
So they don't, not in every tissue, they really migrate only on the vascular network scaffolding. So definitely in the trunk, they do migrate along these arteries, and this is mediated by these chemokines, CXCL12 and CXCL4, that there is quite attraction, and we are trying to figure out a bit more exactly how this is happening. And in the face, they initially start migrating around these vessels, the underneath venous vasculature, but later on, it seems that they migrate much more around cartilage, and it's the cartilage tissue that guides the lymphatics. And I think what really restricts them to go into a lot of space, other spaces, is that there is no space, that there is actually quite a lot of tissue around and they don't have so much choice where they could go.
Thank you. And I also have just a very quick question. So do the single mafba and mafbb mutants, do they still develop the embryos? Just they have faces?
Yeah, so the mafbb mutants, they are adult viable, and we looked also at the maternal zygotic embryos, and they're also totally viable. Whereas the mafbb mutants, they develop only to like 10 days post-fertilization. So they are, so they have other problems. They have otic vesicle problem in development. They have problem in the hindbrain boundaries and so on. So they are, and they don't develop swim bladder. So they cannot swim at the end. I mean, they cannot keep their balance. So they die around 10 days post-fertilization. Yeah.
Okay, thank you very much again, Kaśka.
Thank you.
If any other questions arise, feel free to also write them in the Q&A, and we can, maybe also Kaśka can answer them.
Yeah, I'll be here till the end of the session, so thank you so much.
Thank you very much.